This article, titled Semiautomatic Volumetry of the Temporal Lobes of the Brain and Correlation with Electroencephalography Results in Dogs with Assumed Idiopathic Epilepsy, investigates the potential of using brain volumetry, a technique commonly employed in human medicine, to diagnose temporal lobe epilepsy in dogs. The authors, Paulina Kinga-Drobot and colleagues, posit that this method may be particularly useful in cases where standard magnetic resonance imaging, MRI, fails to detect lesions associated with drug-resistant epilepsy, often linked to temporal lobe epilepsy. The study employed a retrospective observational design, utilizing data from dogs referred for neurological diagnosis between 2016 and 2021. The researchers specifically focused on 31 dogs of various breeds exhibiting dominant temporal lobe discharge, as indicated by electroencephalography, EEG, findings. The methodology involved analyzing MRI and EEG examination results. The researchers developed a novel automated volumetry method, leveraging available canine brain atlases, to assess temporal lobe volumes in the dogs. This method allowed them to calculate the asymmetric ratio, AR, which quantifies the difference in volume between the left and right temporal lobes. The researchers then correlated the AR values with EEG data on epileptiform discharges, EDs, to explore potential associations between temporal lobe volume and the location of abnormal brain activity. The results revealed that 12 out of the 31 dogs, 38%, had an AR greater than 6%, a threshold suggestive of significant asymmetry. Among these dogs, a reduction in temporal lobe volume corresponded with the side of EEG-detected EDs in 7 cases. However, the study did not find a statistically significant correlation between overall temporal lobe volume changes and ED location. Figure 1, a diagram, illustrates the electrode placement on a canine skull model used for the EEG examination. This visual aid clarifies the positioning of electrodes across different regions of the brain, including frontal, central, temporal, and occipital areas. Figure 2 visually represents the process of brain segmentation. The figure provides a side-by-side comparison of brain images before and after correlation with the dog atlas. This comparison demonstrates the alignment of the segmented brain regions with the atlas, highlighting the accuracy of the segmentation process. Figure 3 showcases the results of the temporal lobe matching process. This figure presents a multidimensional view of the matched temporal lobes on MRI, visualized in T1 weighted sequence in 3 planes, along with a 3D reconstruction. The figure emphasizes the precise alignment of the temporal lobes from the MRI scan with the corresponding regions in the canine brain atlas. Figure 4 presents electroencephalographic recordings from a German pointer. The recordings highlight spike activity and polyspike activity originating from the T3 lead, a location associated with the temporal lobe. This figure provides visual evidence of abnormal electrical activity within the temporal lobe region. Figure 5, a diagram, shows the distribution of dogs with an AR above 6%, indicating significant temporal lobe asymmetry. The diagram further classifies these dogs based on the temporal lobe volume, VTL, lateral ventricle volume, VLV, and the side of EDs. This figure visualizes the relationship between temporal lobe volume asymmetry and the presence and location of epileptiform discharges. Figures 6 and 7 and 8 utilize box and whisker plots to depict the distribution of AR, left temporal lobe volume, VTL, and right temporal lobe volume, VTR, respectively, across 3 groups of dogs categorized based on the location of EDs. These figures visually compare the volumetry measurements among the groups. Figures 9 and 10 and 11 similarly employ box and whisker plots to represent the distribution of AR, VTL, and VTR, respectively. However, these figures group the dogs based on the size and symmetry of their lateral ventricles. This visualization allows for a comparison of volumetry measurements between dogs with different ventricle characteristics. The authors conclude that while their preliminary volumetric analysis of temporal lobes suggests volume differences between lobes in some dogs with idiopathic epilepsy, further research is necessary to establish a definitive correlation with EEG findings. They propose that the diagnosis of TLE in dogs based on MRI volumetry, in conjunction with EEG examination, particularly for dogs with drug-resistant epilepsy, could influence the development of new therapeutic options, including surgical interventions. They emphasize that automated volumetric analysis holds the potential to enhance the accuracy and efficiency of identifying subtle structural changes within the brain, particularly in small structures like the hippocampus, which can be challenging to assess manually. The researchers acknowledge limitations to their study, such as the small sample size and the inherent variability in skull anatomy across dog breeds, which may have influenced the results. Despite these limitations, they maintain that their findings lay the groundwork for future research exploring the relationship between temporal lobe volumetry and epilepsy in dogs. They suggest that further investigations with larger and more diverse study populations, along with advanced neuroimaging techniques, could lead to a more comprehensive understanding of TLE in dogs and potentially contribute to improved diagnostic and treatment strategies. From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.